Hi, good evening and welcome everybody to tonight's event. Thanks so much for coming. A great session ahead tonight. My name is Chris Carlton and I'm co-founder of Chen PR. We're a strategic communications firm for high-tech companies and a longtime member of the Enterprise family in Cambridge. Welcome to the program, The Future of the Personal Genome. We've got a great session and a great set of panels tonight. In fact, it's a sellout. So if you could move in a little bit as people come, we should be able to fit everybody in. Before we get to the discussion at hand, I'm going to cover just a few housekeeping items and then get out of the way so you can listen to the brain trust on the topic. First, I want to remind everyone that the forum's mission is to educate, inspire, connect, assist, and advise science and technology-based startups. And the forum is more than just this program tonight or any one program. It covers a wide range of topics, industries, and 4 different program lines, as you see in front of you. The programs really range from pitch and plan feedback sessions to small group seminars for technology entrepreneurs to large community events like we have tonight, all the way to smaller industry-specific groups. In fact, the forum runs more than 70 programs and services a year, and the slide behind me shows what's scheduled for the next couple of months. But if you visit the website, you'll find them all. And a couple of programs that might be of particular interest to the crowd tonight. On March 1st, our Healthcare and Life Sciences SIG is holding a program called Inside the Corner Office, an intimate conversation with Amar Suhani. The session will feature a medical device pioneer, Dr. Suhani, sharing his unique insights on innovating in trying times. I'm sure people in this room have a snapshot about that. Presented in the style of the popular television series Inside the Actors Studio, Dr. Suhani will offer advice on how to adapt to the many challenges presented by the current financing and regulatory environment, including how best to offset risk and how to maximize your chances for success. And on April 26th, we'll be hosting our Innovation Showcase and Celebration 2012, featuring an evening of innovative technology demos paired with some good wine and some good food. So I encourage you to participate in that as well. And actually, we're still looking for some cool companies to participate on the demo. So if you think you qualify, go to the website, download the application, and or talk to somebody here tonight. They can talk to you a little bit more about that. The forum is much more than just the events, as I mentioned. We have one of the largest groups of members in the Boston area. Membership is open to everyone, and members play a key role in supporting the forum, not just financially, but also through their knowledge, their experiences, and their connections. Members are our volunteers, our supporters, our sponsors, our speakers. It really is a pretty wide-ranging group, so if you're not a member yet, you should seriously consider joining. With over 30 years in the space, there are numerous ways to use the forum to build commercial success for your organizations. For the individuals and the companies on the slide behind me and countless others, it's about the advice they got while they're at the meeting, somebody they bumped into at a session, somebody they met during a SIG, that really helped them crystallize what they wanted to do going forward. We want you to be actively involved in connecting with each other because that's perhaps the biggest benefit of the forum is the networking and the knowledge that you can share through that. And we want to know about it. There's something in it for us too. We want to hear your success stories so that we can put them back out to the community and they can learn from them. We're in the process of videoing some of those now, recording them and putting them on the website so people can see them. So if you have a story, an example, a quote, it doesn't have to be long. We'll take short as well as long. But if you think you might have a story to share, please let us know. And we'll talk to you about how to capture that and put it on the website. Like the 28 other forum chapters worldwide, the MIT Enterprise Forum in Cambridge is 100% supported through local programming, fees, membership dues, and support. And the sponsors provide the much needed financial support that allows us to keep the programming fees low. They also provide, equally as importantly, the knowledge, the resources, and the connections to share among the members and with our program teams. Our sponsors are truly invested in the forum's mission and also have long been invested in helping build the ecosystem in the Boston business community so that it thrives to the level that we all want it Tonight, the forum would like to give some special thanks to two members of the Innovation Series Committee, Barbara Ewan of Trent PR, one of my partners and a longtime friend, and Laura Kachanova of Scratch Media and Marketing. Ladies, hands up, please, so you can know his name. And following tonight's session, we encourage you to join us at a networking reception at the R&D Club on the 4th floor of the Status Center. Tonight's program is sponsored by WilmerHale. I'm pleased to introduce Joshua Fox, partner at WilmerHale, to say a few words. But before I do, I want to say that Josh and I are an example of the connections that you can make through the MIT Enterprise Forum. We met each other through an acquaintance and had lunch a while back. Our respective crazy schedules have conspired against regrouping. But tonight, I get the opportunity to see Josh again, so it really does happen. Josh?
Thanks, Chris. Thanks for the warm welcome and good evening, everyone. On behalf of WilmerHale, I'd like to thank you all for being here. I would also like to express our appreciation to the MIT Enterprise Forum for its dedication to innovation and technology, which brings us together this evening. The Enterprise Forum has proven to be a pillar of the entrepreneurial community here in Boston, building connections among the region's most promising technology and life sciences entrepreneurs, and delivering timely content on emerging and growing industry sectors. This evening's panel on the future of the personal genome certainly answers that call. Recent developments in genome sequencing have produced exciting new opportunities and technologies, and thus, to no surprise, new startups in the growing personalized medicine area. My WilmerHale colleagues and I have had the privilege of working with many of these innovators, life science companies of all sizes, to advise them through a myriad of legal, intellectual property, and regulatory issues. The scientific breakthroughs and the concepts that we'll hear about this evening, and some that may be just the seeds of game-changing ideas in the audience, challenge us to stay current and stay curious. It gives me great pleasure to welcome our esteemed panelists, and thank you again for being here.
Good evening, everyone. Very warm welcome to you. Great to see such a fantastically large and diverse audience for this MIT Enterprise Forum event on the future of the personal genome. I am delighted and honored to be invited to be the moderator of this evening's event. My name is Kevin Davis. I'm the editor of BioIT World, a web portal with news and information on technology and life sciences, and also the author of the $1,000 Genome. I promise not to plug this overtly during the rest of this evening. But if I don't see a sharp spike in sales on Amazon, I'm going to be bitterly disappointed. If I can't sell to this crowd, then there's no hope. I've had the pleasure of knowing and working with everybody on this panel on various occasions, but not in this particular combination. And I commend the organizers of this evening because they pulled together a fascinating group who all come at this field of genomics and personal genomics from very different avenues, from computer science, from engineering, from physics, from medicine. And so I think you're going to hear about a very fascinating range of opportunities and challenges in using this phenomenal breakthroughs that we're seeing in genome sequencing and personal genomics. I'm going to introduce our four guests in just a minute. I'm going to first take the liberty of taking 2 or 3 minutes to just quickly bring us all up to speed. I think many of you are familiar with what we're going to be talking about, but I know that a few people probably aren't, and but are obviously aware and interested in following the news, which we can't escape, about advances in DNA sequencing technology, medicine, personalized medicine, all of these buzzwords that are floating around the scientific and mainstream and business press, and wondering what it all means and what's in it for us. I think the first personal genome, I think opinions vary, but if I had to hazard a guess, I would say probably Ethan Hawke in the film Gattaca. which you may remember came out in the late 1990s, and I'm sure if you have seen the film, which I'm sure many of you have, you'll remember that the perfect specimen, Uma Thurman, taking was it a fingernail or a bit of hair from Ethan Hawke's keyboard and taking it to her CVS DNA sequencing while you wait dispensary, and being rather disappointed by the results that came back. Of course, a notable landmark in this field was the completion of the Human Genome Project, done about 10 or 11 years ago. And an event that really spurred that event on was obviously the creation of a company by Craig Venter in 1998 called Celera Genomics, and they published in the same week, in February of 2001. And everyone thought at the time that Celera had sequenced 5 anonymous individuals under very rigid IRB rules, only for Craig to go on national television a year or so later and announce that in fact his had been sample number 1. I'm not sure how it got there, but it did. And he said to the rest of the field, you know, don't be such a hypocrite. If you'd been in my shoes, you would have done exactly the same thing. So while Craig gets credit perhaps for being the first fully sequenced individual, remember that was done using first-generation sequencing, a good old-fashioned, still used method of Sanger sequencing. And in about 2005, 2006, we saw the first commercialization of the second wave of DNA sequencing technologies, miniaturized, multi-parallelized, very much faster and very much cheaper. And in May of 2007, James Watson, of course, the man who co-discovered the double helix structure of DNA, was presented with his digital genome on a portable hard drive by one Jonathan Rothberg, who had created a company called 454 Life Sciences. That was the first commercial next-generation sequencer. The cost of that sequencing project in $2007 is put at about $1 million. It didn't leave much for gift wrapping. You see the chintziest of red ribbons tied around that contraption, and Watson's sequence was published a year later. Jim was guest of honor at a gathering that George held not too far from here a couple of years ago, in which he brought together more or less everybody who had been sequenced at that point. This was in May of 2010 at the inaugural GET conference. And you see on the top right a number of members, inaugural members of the George Church Personal Genome Project. We'll hear a little bit more about that, or a lot more about that, in just a few moments. That's certainly one, and an existing current way that you can get your genome sequenced. Another was evidenced by another guest at that reception, Ann West, a Californian teenager who had her genome, along with the rest of her family, sequenced by Illumina, which rolled out a genome sequencing service. And for George's, to get onto George's program, you had to pass an exam. For this program, you had to write a pretty steep check, although the zeros are falling off very quickly. And you also need to provide a doctor's note, and this is the note that Ann West provided. It says blood and saliva for personal genome sequencing by Illumina. The cost at that time was, I think, $40,000 per person. It's now somewhere under $10,000 and continuing to drop. Another way, of course, in the last few years that people have been getting information about themselves, about their genetic identity and their health predispositions or illness predispositions, is through direct-to-consumer genomic services such as 23andMe. I suppose one of their most famous subjects is Sergey Brin, the husband of company co-founder Ann Wojcicki. George is an advisor to the company, who learned that he carries a mutation by browsing his raw genotype data, learned that he carries a mutation in a known Parkinson's risk gene, putting his risk somewhere around 50%. He has a known family history of the disease, so this was not altogether a surprise, but still pretty bracing to see this. And it raises the concept of the personal utility of this information. It may not have immediate medical utility, but sometimes that may not be what people are necessarily looking for. We might talk about that as well. And 23andMe has now branched into DNA sequencing and now offers not the $1,000 genome, but the $1,000 exome, which is what we call all of the genes sort of stretched end to end, cutting out all of the junk DNA that we don't necessarily know a whole lot about at the moment, although we're learning more and more. And that's currently available to existing subscribers for about $1,000. I think one of the more remarkable trends in the last year has been seeing this move into the clinic. And this is one of the more famous sort of early poster children of clinical genomics or personal genomics in the clinic. This is a young Wisconsin boy named Nicholas Volker, who's had quite a bit of press in the past year, including, see the URL, bottom left, this is will take you to a Pulitzer Prize winning series of articles in the Milwaukee Journal Sentinel recounting this boy's diagnosis with a mystery inflammatory intestinal disorder, which finally his physician just couldn't, and tired of reading him in for yet another surgery. He had something over 100, when finally his physician contacted a genomics expert at the Medical College of Wisconsin named Howard Jacob and said, "Do you think we can sequence this boy's genome? We just don't know what else to do." Jacob's team did that. They found a mutation in a known gene, and you can read the details of this series. He had a bone marrow transplant, which almost certainly would never have been authorized without that genetic diagnosis, and as a result, he is now home. I wouldn't necessarily venture to say he's out of the woods, but at the age of 7, he finally went trick-or-treating for the first time and is eating solid food, and it's a pretty, so far, a very good story. This group is one of now a growing number of groups around the country that is taking whole genome sequencing and utilizing it in a variety of clinical genomic situations on a more and more routine basis. And it's leading some people who were once skeptical of the power of whole genome sequencing in a medical context to say, to utter words that they probably wouldn't have dreamed of saying even just a couple of years ago. David Valley is one of the more respected medical geneticists in the country, and at a recent conference he said, "I believe that the standard of care will be sequence once, read often." And I was quite surprised and shocked to hear somebody of David's stature actually sort of drinking the Kool-Aid, so to speak. And some people on the panel and in the audience may fully endorse this view, and others may take issue with parts of it, or believe that we're getting a little carried away with ourselves. So these are some of the things we're going to discuss. And my final, I think, introductory slide, just to touch on the ever-growing wave of new technological developments. Many of you will have seen just in the last few weeks the announcement from Life Technologies and their subsidiary Ion Torrent that their new machine, known as the Ion Proton, because what this is, when you strip it down, is really a glorified pH meter, is a machine that will deliver the $1,000 genome by the end of this year or the beginning of next year. And if that is indeed the case, and they can do this in a very short space of time and to a suitably high degree of accuracy, then I think that will put them in a very interesting position, maybe even pole position, to claim a share of the $10 million X Prize that will be given for genomics. That will be given one way or the other when the competition is held in January of 2013. Maybe one of our guests will have something to say about that before all is said and done. So we can see we are getting very close to this era of very cheap, accurate, affordable genome sequencing, which then begs the question, what are we going to do with that information? So that's a little primer on personal genomics, and the way we're going to do this evening is we're going to discuss a number of topics here on the stage for about 55 minutes or so, and then we have the luxury of a lot of time to take questions from the audience, at least 30, maybe 40 minutes, and I'm sure that you're going to be thinking up far more varied and provocative questions than I can possibly dream up. So please be thinking of what you want to ask the panel. And indeed, if during the conversation you just have something profound that you wish to share with the entire audience, I'll look up and I might call on you. We've asked each of our 4 guests to just introduce themselves for just a few minutes, show a slide if they so choose. 3 of them have offered to share slides. This is George Church's slide. So we're going to start with George. As I said, what's great about this quartet of speakers is that they each come from very different backgrounds. George really started life as a fascinated in all things computing. Famously got kicked out of the Duke University PhD program. He proudly shows his eviction letter on his website. Sweet-talked his way into a position with Wally Gilbert and became one of the very early sort of thought leaders in genomics and the Human Genome Project. Virtually every company working actively starting up in this space has his fingerprints on it, either as a co-founder or as an advisor. And it's a great pleasure. We really couldn't have this session without him. So George, I'm going to invite you to the welcome, George Church.
Thank you very much. When faced with the prospect of one slide, I really couldn't beat the minimalist approach here. I hope my talk is not content-free. A few corrections for Kevin, well-intentioned corrections.
You always do this, George.
Different corrections each time. He's a quick learner. So it's not $10,000. The street price is $4,000 from two different companies, Nomi and Illumina. And the $1,000 projected price is projected by two companies, both Life Technologies and Illumina. And a very important aspect is the accuracy of these genomes, but the most important is interpretation, and that's mostly what I'll talk about. The accuracy, I think we'll see an announcement soon of 10 to the minus 7th. That's one error in 10 million base pairs. It's about 50 times better than anything previous, and that's from Complete Genomics. And our group we just submitted a paper on that. So interpretation. To do interpretation, I would argue that we need a cohort. We need a cohort which is available, where the information is available to everybody in this room and internationally. And it is not just a tiny slice of the genome. So some of the companies that were mentioned, Nanogenix, 23andMe, and so forth, they're giving you a fairly small slice of the genome. There's a lot that can be done with the whole genome. And what can be done is not merely pharmacogenomics or cancer or entertainment. It is just about everything in medicine. So how many people here, just a show of hands, are absolutely convinced that they do not have hypertrophic cardiomyopathy, which is an adult onset disease that has really little to do with other heart diseases? Show of hands, who knows? Okay, that's a little disappointing. So how many? My count was 0. So how many people here have had some aspect of the genome analyzed? Let's ask that. Okay, so there's a fair number of hands that went up. That means that who's ever analyzing your genome is not reporting your hypertrophic cardiomyopathy. But it can be done, and it's routinely done in a variety of medical genetics centers all over the world. And what's happened is the kind of the direct-to-consumer and even the genome-wide association studies, which have dominated the news, have convinced us that genetics doesn't work. And I'm here to remind you that genetics has been working quite well since 91 at least for people with familial histories. But the thing is, a lot of genetic problems, you're the first one in your family. And so you can't just wait and say, okay, we're only going to treat people who have a father, mother, or other relatives. The cost of the genome now is on the order of the same price as getting a piece of it. So Myriad offers BRCA1 and 2 genes for about $3,500. Nanogenix, a few SNPs that are not quite as actionable as Myriad's great BRCA1 and 2 for $2,500. So you can get your whole genome by the end of the year, $1,000, but right away for about the same price as 2 genes or a few SNPs. So you should. If you can't save $4,000 over your lifetime in healthcare costs by creatively using your genome, you, pardon me, don't belong at the MIT Entrepreneur Forum here. Personal Genome Project, just quickly there, that is what is the data set that we can all share. It is the only project that I know of in the world. It's sort of the heir to the Human Genome Project, which is about an imaginary hybrid genome, a haploid genome. Even Craig is not haploid, as far as I know. Actually, he later, 3 years later, published his diploid genome. There is no project other than Personal Genome which allows you to share data on genomes, environments, and traits, or pretty much any 2 of those. Anything that's sufficiently identifying about a human being is locked away in a vault, which is comparable to Fort Knox, called dbGaP. How many people here have actually used data from dbGaP? 1, 2, 3. That's better than the number of people who know about HCM. That's great. But anyway, it's hard. Do we agree on that? It's pretty hard, yeah. I see the nodding heads. PGP is not as hard. You click on it, you got it, okay? It's consent. Furthermore, a lot of shared resources are not consented for commercial use. If you want to use a cell line, good luck. But the Personal Genome Project, because it was supported privately, it is Creative Commons zero license for all the data, which means you know that you're safe if you're a company. And you can patent anything in there you want. You would just have to be the first to make any value out of it, but it's free for you to patent if you have ideas based on it. And you can use the cell lines any way you want. So if you want to clone me or make a transplant based on me, I'm not going to come in for a piece of the action later on. Okay? So those cell lines are distributed by Coriell and by the Personal Genome Project directly. So it's a biobank resource. I don't want to go on and on about it, but I wanted to make that one thing clear to stimulate conversation, and I'm really looking forward to the conversation.
Thank you, George. Colin Hill will be our next person for you to meet. He is the CEO and co-founder and president of GNS Healthcare. Colin is a physicist by training. I first met him about 10 years ago when he was just starting Gene Network Sciences and building very advanced in silico models of cancer cells, and I didn't know whether he was brilliant or crazy, or maybe a bit of both. But the new iteration of GNS Healthcare aims to help their clients discover the knowledge that they need to match patients with treatments that work. So kind of the interface of bringing personal genomics to personalized medicine. So let's welcome Colin Hill. Colin.
Thanks, Kevin, and thanks to the organizers for having us here. I think I have a slide. This is the next slide. Okay, so I'll tell you who's in there. So that's me on the left, if you didn't recognize me. And unfortunately, the person on the right is my cousin. This picture was taken 3 weeks ago in Ottawa, Ontario, Canada. And that was around 10 days after I got a call from my mother telling me that my cousin, TD is her name, that was her nickname, was diagnosed with stage 3B cervical cancer. At least that's what they thought. This eventually became stage 4 cancer, and she's currently in the ICU right now. So she's 43 years old, otherwise rather healthy. And so the reason why I've made this the picture to talk about personalized medicine and the implications of the $1,000 genome is because this is, for me, a time when this technology and all of these developments really count. So after 11 years since we started this company, with Abe Khalil who's in the front here, which really aimed to pioneer the applications of machine learning and modeling and simulation running on very large supercomputers to take multiple layers of data, so DNA sequence data, but also expression data, other molecular profiles with clinical outcomes, and even now working on the real-world outcome side with health records. The goal was to reverse engineer models of these systems and ultimately get a predictive understanding of how to intervene in the care of a patient. That was the goal 11 years ago when we started. That's the goal now. The problem we're facing here is with my cousin. Now this is not working with a pharma company trying to stratify patients in a trial from 250 patients worth of data with outcomes and such. This is now my cousin. This is truly an N of 1. The problem is we don't have the full data in order to solve this. While because of the field we work in, I'm able to call up a lot of the big cancer experts in the world from people who have the NCI, that's the National Cancer Institute, or people at MD Anderson. No one can really help my cousin at the moment. And so where we've gone to, there's not zero hope. This is not just a completely sad story. The story is still going on, to be clear. This is 5 weeks in. We actually had her tumor biopsy sent to Foundation Medicine. You're going to hear from Mike Pelini later this evening, with the hope of finding therapeutically actionable genetic aberrations, deletions, mutations in her tumor genome that could lead to a therapy. Right now, after standard chemotherapy, cisplatin and paclitaxel, not a lot has happened. Her chances of survival are really, really low. So the only hope is to ultimately use advanced profiling to get a second chance, of finding a therapeutic intervention. Now, in her case, given just her reaction to the chemo, the chances aren't great. But we are absolutely going for it. And what I guess it's really taught me, having been in the industry for some time now, is really the fact that we are soon going to have high throughput molecular profiling, not just of the DNA sequence, but of entire tissues, together with longitudinal health records. There's now the chance to really unravel the key questions and using causal inference, get at the underlying mechanisms of a disease, such that one can take data from a given patient, like my cousin, plug it into a computer model, and test out many different interventions and determine what the best one is. It's a vision that's crazy ambitious, but it's a vision that's actually starting to become real because of advances in molecular profiling, but not just molecular profiling, the availability of longitudinal health records. You need data that shows, did the person live, die, how long did they survive, and also data around the process. Of course, DNA sequences are powerful, but they don't change from the time we're born, right? Unless it's cancer. So I'll just end with, I guess, our vision, my personal vision, which our company certainly embraces, is that with increasing data liquidity, and what I mean by that is the fact that electronic medical records, longitudinal claims data, more clinical data sets are coming online associated with samples, such that a $1,000 genome, and soon to be even cheaper than that, are going to be really a part of a revolution that's going to enable personalized medicine to really happen. It wasn't possible 10 years ago when people were first talking about it. It's not quite possible today, but we are indeed getting there. And so we see really a convergence of innovations in artificial intelligence, supercomputing, and data profiling being really the key to a new generation. Thank you.
Colin introduced you to Foundation Medicine, and our third guest is Michael Pelini, who is the President and CEO of Foundation Medicine. Michael is a physician who's spent a decade or more in working and starting various biotech companies, and now leads, I think, one of the most exciting ventures in molecular diagnostics devoted to the cancer space. It's the lead story, coincidentally or not, on bioitworld.com today. Michael, welcome, please.
So first of all, I'd like to thank Kevin and Barbara for the invitation to speak tonight. So we're starting off with a couple of talks here, a couple of introductory comments on oncology. And I know tonight's discussion is supposed to be about the personal genome and what can we do with it. I think looking at oncology, looking at cancer for a moment, gives us actually great insight into where the world is going to head over the course of the next 10 years. The practice of oncology is changing, and I would say that the practice of oncology is actually in the process of going through a revolution today. And that's a great thing for all of us. It's a great, as humans, as oncologists, as patients, as family members of patients. And I think it was really helpful to have Colin's comments before I talk. Before I jump into it, though, I want to ask the question, how many people in this room have read the book, The Emperor of All Maladies? So a fair number. Okay, so maybe 10% of the audience, 20% of the audience. This is actually a slide, and so this is the black bile, otherwise known as cancer, that the book often refers to. And it's a black slide, so you're not missing anything. It's a little bit different from George's slide in the sense that it is actually a slide. And not that long ago, this is where we were in trying to understand, what is this massive tissue? It was a black box to everybody. And then we got to the point where a pathologist could take a slide, look at the morphology, look at the slide and say, this looks like lung cancer. This looks like breast tissue, and these cells are have changed, so this is breast cancer, and so on. And the world started to evolve. Unfortunately, and I guess unfortunately, it evolved relatively slowly in the diagnostic world. And then we started to learn a little bit more over the past decade in particular about the application of these molecular techniques into the world of oncology and specifically with diagnostics. So about 5 years ago, we started to recognize that, hey, there's an important marker called EGFR, thanks to some wonderful discoveries here in Cambridge as well as in New York City. And we understood that this is a change, a molecular change, in this case, lung cancer, that was actually targeted by a therapeutic that was on the market. So that's step number one. We started to get a peek into this black bile, into this cancer. Then we started to learn more. A few more targeted drugs entered the market over the course of the next few years. We're starting to get a bigger picture, a grander picture, and so on and so forth. Where we are today is that we actually do have the ability to understand all the molecular changes, the key molecular changes, the driver, that are actually causing many of the cancers. And most importantly today, that we're not just generating this information for the sake of it. There are a lot of folks out there doing research, and that's a great thing. What we care about as a company, and I think what many of us care about as clinicians, is so what? What can you do with all this information? The reason that oncology is going through a revolution today is that we're not only generating information, we're actually generating information that's useful. And I got a hard time from Jamie for using 3 slides today, so I'm obviously compensating for something, but I want to make a point here with 3 slides. So this is an example of actual data that we have generated. And it's this data is based on information, our sequencing that was done on approximately 90 patients with non-small cell lung cancer. We found alterations in approximately 31 genes in those patients. Now, one point that I want to make here is even though we found alterations in 31 genes, and it's difficult to see on this slide, in fact, only 3 of these tests, only 3 of these changes would be routinely looked for if you're a cancer patient today who has non-small cell lung cancer. So you would only look for 3 of those changes. And if there are oncologists in the audience, when I say routine, you're chuckling because there's really a very small percentage of the patients that even get all 3 of these tests today. But what's more important, let's go back to what can we do about it. 31 genes altered. However, in 18 of those genes that are altered, we can directly tie therapeutics that are on the market or therapeutics that are in clinical development to those genomic changes, to those molecular changes. That is a world from where we were just 5 years ago, and it's only the tip of the iceberg as compared to where we are going to be in the next 5 and 10 years. So this information can be generated today on cancer tissue. It can be very meaningful today. So even if we could do it 5 years ago, even if we had the resources and we could run these tests, we could run these next-gen sequencing-based tests with great efficiency, it still might not have made sense to do it because we didn't have any therapeutic interventions. And then finally, I just want to give you a sense of where this is going. And this is not 2015, 2020. This is 2012. This is 2013, where we and perhaps others are producing this information so that an oncologist in his or her office can pull up the information from that very patient, can either on a paper report have summary information that they can understand, that's actionable, that they can interpret in 30 seconds and say, aha, I don't only have information, but I know what to do with this information, or they can do it on a web-based tablet, where if you look at this, and I'll walk you through a couple components here of this slide. So your tablet, in the top left, you have all the information on the patient, the demographic data, the information on the physician, the initial diagnosis. In the top right-hand section, you have that early diagnostic data, that morphology interpretation by the pathologist. As you scroll down to the middle left box, those are your genomic changes those are the genomic changes that were found in the cancer tissue itself. You have the ability to drill down, and if you want to go and better understand each one of these alterations, you click on a link and continue to drill down you're going into the public domain now to access that information. On the right-hand side of the middle, you have the therapeutic agents that are targeting those changes, and those are agents that are available on the market today. On the bottom left, you now have a link, whether it's via Google Maps or just a listing, of clinical trials that are targeting those molecular changes. And so if you don't have a therapy that's on the market, you have access to the trials around the country, so you and your oncologist can look at these trials to see what is appropriate. This is not 5 years away. It's happening today, and it's going to continue to evolve. And so I think this is our earliest glimpse into where we are going with the personal genome. I think there are a lot more fun things that we can talk about, like some of the information that you glean from 23andMe, and I don't mean that in a negative way, I mean that in a positive way. They'll provide important information, so it's not all cancer, but I think cancer is just a great representation of what's going to happen in the medical world, and it's actually happening today, but we're just at the front end. So thanks for allowing me to take a few minutes.